So yeah, indeed. What I'm going to be doing today is giving you a bit of an overview of Fluidigm has to offer indeed in the spatial transcriptomic realm, but also more broadly in the spatial biology realm, I would say. And so, of course, I mean, the whole theme and topic of today's conference, again, thanks very much for the invitation, is of course talk about spatial transcriptomics and how really this is feeding into this huge revolution that is spatial biology. And so, of course, again, today's topic is spatial transcriptomics. We are talking a lot about it, and we've really seen how, even though we're getting closer and closer to it, the bulk of today's technologies are really focused on really being able to apply spatial transcriptomics still more to the analysis of areas of tissue or multiple cells at the same time to do perform either targeted or really whole transcriptome-wide assays or even gene expression assays, even though, as we saw, we're getting closer and closer to really being able to apply the real and true single cell resolution. At the same time, we also know that there is another, let's say, battery of technologies that are out there on the market, which are really propelling the spatial biology revolution forward. And these are more what we can think of as highly multiplexed imaging technologies. And so while it might be a bit of heresy to talk about this in the Spatial Transcriptomics Congress and Forum, these technologies, of course, use in most cases antibodies to be able to go in and actually look at predefined targets and panels. But the big advantage of using and incorporating highly multiplexed imaging assays within the spatial biology realm, of course, is to be able to go in and really use the technologies to really easily, and I think also very efficiently, really identify which cell types can be present within any type of tissue section. And by this then, be able to quite easily go in and really perform all those studies which involve understanding how and if the actual composition of a tissue section with all of its cells and where these cells are actually located actually can go in and give us more information on what's actually happening either from a biological perspective or in that disease state, or of course, how these different cells can be responding to different treatment options, et cetera. And so with that in mind, I'm here to talk to you about how Fluidigm is playing a role in this, because fundamentally, we believe that we're playing an interesting role again in the spatial biology research arena, because by fundamentally being capable of placing any type of tissue section on any classic glass microscope slide, we can now take that slide and either in the realms of highly multiplexed imaging, use our Hyperion imaging system, which is fundamentally a technology which allows for an easy multiplexing in the realms of immunohistochemistry of more than 40 plus markers, again, with a subcellular resolution to really visualize where all these different markers are on a tissue section, to really be able, again, to rip out all those cell types and understand not only the function, but where they're localized. And I'll also be talking more about how this technology can also be used not only in combination with spatial transcriptomics, but also to be able to actually be used to even get a simultaneous multiplex RNA and protein readout on the same tissue section at one given time. And of course, in the realms of spatial transcriptomics, what I'm going to be talking about is how we are also participating within this research area directly, because with our up and coming Acculift laser capture microdissection system, we also now are allowing researchers the possibility to be able to go in and either microdissect areas of tissue, but even also single cells, rip them out, and then fundamentally, which I think is something quite unique, is fundamentally do whatever you want with them. Because once you've actually ripped out that piece of tissue from the slide, you can go in and perform any type of downstream assay that you are interested in. And we also have, if necessary, but you're not forced to or obliged to do so, also have downstream technologies which can really allow you to go in and perform these sort of either gene expression or targeted NGS or even whole RNA-seq assays in a very cost-efficient and high throughput manner, if need be. So with that in mind, I'm just going to be giving you a brief introduction into these technologies. I'll be starting with the Hyperion imaging system, which is a technology which, again, as mentioned beforehand, very simply allows you to do a highly multiplexed immunohistochemistry experiment. This technology is based off of Cytov technology. And for those of you who do not know what Cytov technology is, it's fundamentally a technology which was released more than 10 plus years ago now, which really overcomes all of the classic limitations that we, of course, all are aware of when using fluorescence-based modalities and being able to get, multiple markers to work with different fluorophores in any given tissue section. mainly because of autofluorescence and, of course, overlapping excitation emission spectrums, and how we fundamentally now take all those regions which we can use to perform the immunofluorescence approach, but now instead of tagging these with fluorophores, we now go in and tag these with highly pure, highly purified metals isotopes. And so by doing these now, we really get these separate and distinct signals, which is what allows our customers to be able to go in and, again, quite seamlessly create panels from more than 30, 40 plus markers to be able to visualize these again on any one tissue section. And with that in mind, as I'll be showing you later, this whole workflow is really quite simple because, again, this technology works on fundamentally any type of tissue section. So this can be either formalin fixed paraffin embedded tissue sections, frozen tissue sections, even cell smears, if you want to. And what you're just fundamentally doing is taking that tissue section, placing it on a classic glass microscope slide, and you're just performing a classic immunohistochemistry experiment, in which big differences I'll show you later. You're just taking all the antibodies, literally throwing them all on top of the glass slide at the same time. And then we're capable of going in and ripping out these images at a subcellular resolution and generating images like the ones that you see here, which now it's kind of like looking at a classical image from an immunofluorescent microscope, but with the possibility to really now be able to go in and rip out all of these different markers with the subcellular resolution to really understand all the cell types that can be present on any given slide. So that being said, very briefly, as I mentioned beforehand, this technology really allows you quite easily to go in and create these large panels. Something I just want to briefly mention is that we already have a catalog readily available that works on both human and mouse FFPE and frozen tissue sections, which you can go in and mix and match and use. We also have ready-to-use kits. But again, something which is quite important, I'll show you a real-life example of this, which has really made this technology become so successful, is the fact that if you do want to use such a highly multiplexed imaging approach, it's quite easy to go in and really take fundamentally any marker of your interest or antibody of your interest, but again, I'll show you later, it's not only antibodies, and tag it with the metal of your choice by using our labeling kits, which really allow you to go in and customize your assays as you see fit. And very briefly, again, as I mentioned beforehand, I think what is the beauty of this technology really lies in its simplicity, in the sense that it's, again, just fundamentally immunohistochemistry. You're just taking the slide, treating it as you would in any classical IHC workflow and protocol. But the big difference is that it's actually quite simpler to a point, because you're just literally throwing all the primary antibodies conjugated with these metals on the slide at one time, then taking that slide, plugging it into our machine, and you're ready to go in and generate these images. And the use of the machine itself is also quite simple, in the sense that fundamentally it doesn't really require any microscope experience, to be honest, because all you're doing is really switching it on, introducing the sample inside the machine. As I think has also already been introduced beforehand by Fadi, like with the Juno, you can also post-stain the slide, for example, with immunofluorescently tagged antibodies, in case you would need to go in and really be able to specifically localize regions of interest that you want to go in and acquire specifically on the Hyperion imaging system. You select those regions of interest, then you click play, and the machine automatically goes in and actually starts acquiring all those different regions of interest and automatically generating these images, which can then be analyzed downstream. And something that I just want to briefly mention, which I think is quite important, is that, especially in the realms of spatial transcriptomics and all these technologies that we're talking about, is that the data sets that we generate actually with this technology are very, very small. We're talking about approximately 150 megabytes for a typical region of interest of 1 square millimeters, which for about like 30, 40 markers, which means that these data sets are really easy to handle and can be analyzed directly after any single run has actually been finished. And it's really been in the simplicity of this technology that I believe that its success can really be found, because you can see that the technology has been now used in more than 100 publications worldwide. You have more than 100 systems that have been installed pretty much all around the world. About half of these are actually in Europe, including, of course, Oxford. And I think something that can also speak nicely about the rapid implementation and panel design and creation of this technology can be seen, for example, within this COVID pandemic, in how, for example, 4 different COVID-19 publications specifically using imaging mass cytometry with the Hyperion imaging system actually came out. And one very interesting one that came out is one that was recently published in Nature by the Schwartz and Elemental Labs in Cornell, in which they were actually capable of going in and taking lung biopsies from different patients, which unfortunately, of course, died due to COVID-19. Compare these to actually non-COVID-19 deaths, specifically also deaths which were also induced by acute respiratory distress syndrome, but by other infectious diseases like bacterial pneumonia or more of the type of infectious diseases mentioned beforehand. And by actually going in and taking these sections, and now actually going in and applying like a 38 antibody panel, they were capable of not only identifying all these different unique immune and also epithelial cell types that, of course, actually reside in the lung per se. But something which was interesting, and I think also Fadi was referring to this beforehand, is that they were also capable of going in and really for the first time specifically understanding which are the cell types that are actually being infected by SARS-CoV-2. And in this case, by taking an antibody, which reacts in immunohistochemistry to the spike protein of SARS-CoV-2, they were capable of tagging this antibody with one of our labeling kits. And as you can see here in green, they were capable of specifically going in and showing, for example, that it's the alveolar epithelial cells that seem to be the main target of infection by SARS-CoV-2. And by uniting all this information together with all the other like immune cell infiltrate information, they were really capable of getting a much better understanding of what is really specifically going on within lung biopsies of these deceased patients. But as mentioned beforehand, I also want to briefly touch upon how this technology can be used also in the realms of spatial transcriptomics, and it can also be used to be able to look at things other than antibodies. There's already quite a few publications out there, and this was the first one, which I believe is quite nice because it really gives you not only the proof of principle data, but really gives you a biological insight on what can be done when you do this. This technology which shows how you can use mRNA fish probes, like the ones from RNA Scope from ACD Bio. And instead of doing a classic fluorescence-based approach to look at these, actually tag these probes with our meta labeling kits, and now be able to actually look at with subcellular detail at mRNA targets, but also antibodies, again, all on the same tissue section. And in this case, they actually applied this technique to look at different chemokines, for example, which can be very difficult to look at the antibody-mediated techniques. And in the case of breast cancer, really, they started to see how for example, CXCL10 actually really form sort of patches in which T cell abundance can be found specifically in specific types of breast cancer. So this was a bit of the overview on the highly multiplex imaging, but of course, I really also want to come here and talk to you about this up and coming technology, which is the Accurate Laser Capture Microdissection Machine, which is fundamentally a new laser capture microdissection machine, which now will allow customers to be able to go in and with quite a very simple and easy format to go in and either go in and acquire areas of tissue or even individual cells at any one time. And once you've actually done that, analyze those areas or individual cells with fundamentally any compatible downstream assay of choice, depending on how that tissue is actually processed upstream. And so, for those of you who might not be aware of literally how laser capture microdissection works, it's actually quite simple. All it is a microscope in which you place a slide inside, and via laser-based system, you can go in, identify the target cells that you're interested in, cut them out, and then these cells will just literally then be picked up, placed onto a cap, into an Eppendorf tube, with which you can then do, as mentioned beforehand, pretty much anything that you want. This technology will come with pretty much everything, of course, that is needed to make it tick, including, of course, all the different slides that can be necessary for it, and even the purification and molecular analysis reagents to make this technology work, even though, as mentioned beforehand, while we sell these kits, you're not forced to use them. You can use any other downstream kits or assay of choice that you might already be using within your transcriptomics readouts. And to show you how this technology can actually be incorporated, which I think is also quite important when we're starting to think about spatial biology into an actual digital pathology workflow, because now that we're actually looking at pieces of tissue, it's important for us to understand what areas of tissue might actually be interesting for us, et cetera. We already know that, for example, laser capture microdissection is being used routinely, I would say, even in the clinics. And while this is a simplified diagram, it can work like this in the sense that a clinician says or a pathologist says, okay, you have this piece of tissue. Now I need to be able to efficiently identify which are the areas of interest for me. And this can actually be done quite easily with this up-and-coming system, because fundamentally, this technology will allow you to place a slide directly on the laser capture microdissection system itself, do a whole scan slide. And once that's actually done, that image can be uploaded directly to a cloud-based system that we can manage. And once that's done, you can go in, of course, zoom into the image as required, annotate that image as need be. And this can be done, of course, also offline by a pathologist, so nobody that's actually anywhere close to the machine itself. And once that's done, all that information can be then placed into a virtual notebook in which you can go in and again, specifically select those ROIs of interest, which can either be, again, either areas or even individual cells. You can then take all that information, export it directly to the machine, in which you can then go in and just fire the laser, in which you'll actually be removing all these individual sections, be placing them on the caps as mentioned beforehand. And then you can go in, and once you actually have all of those caps and tubes, you can go in and again, purify the material as required, either for DNA, RNA, even protein if need be, and again, do the molecular analysis that is required. And again, while you can fundamentally do this with fundamentally any assay or kit that you want, if you do actually require it, what we also already have in Fluidigm is a portfolio of different regions and also instruments which can allow you really to go in and perform seamlessly any type of, for example, targeted NGS or even whole RNA-seq library prep. To do this, we have the Juno, which is fundamentally an NGS library prep instrument, in which you can go in and create any sort of custom targeted NGS panel or use pre-configured panels that you can also modify as need be. And what this really allows you to do is, again, take these pre-configured chips that we have, which I'll be showing you later on, load them up, and then everything which is the library prep, again, for either targeted NGS or even whole RNA-seq assays, total RNA-seq assays, can be done completely and automatically by the Juno machine, which means that all you have to do is literally go in and harvest the cool barcoded amplicons for your downstream sequencing. And this can be done by, again, using these wide variety of chips that we have at our disposal, which can allow you to go in and pool either 48 samples at a time and go in and look at more than 4,800 amplicons per sample. You can either look at 192 samples and look at up to 2,400 amplicons per sample, all at one time on one chip at any given moment. You can do this also in a very flexible format, for example, with this one chip that we have, which is called the 886 chip, which is kind of like having 6 chips in one, in which in case you have, for example, a wide variety of different targeted NGS panels, you can load 8 samples per partition, so you can have like 6 independent targeted NGS panels on this assay for these 8 different samples at any given time. And as also mentioned beforehand, we also have a chip which can allow you to completely automate the whole RNA-seq library prep process for 48 samples at a time, which we know can be a very, very labor-intensive process. And so with all that being said, I just wanted to quickly finish with showing you actually one paper which shows how I think these two technologies, the highly multiplexed imaging and also spatial transcriptomics, can be united in one. This is a recent paper that was published now in Cancers. And it really shows this, what they call this nice spatial imagenomics pipeline to be able to identify prognostic biomarkers associated with the ovarian tumor microenvironment. And fundamentally, what they did here is by taking quite a large cohort of samples, because I think the nice thing about applying these techniques is that they're also very cost efficient in being able to really increase the N number for the sort of experiments that you're doing. And by performing, on one hand, imaging mass cytometry, they were capable of going in and again, identifying all of those different cell types that are present in these different ovarian tumor samples. But even more importantly, identifying not only the cell types, but which were the kind of cell-cell nearest neighbor interactions which actually predict the best survival amongst these different patients. And then at the same time, also by doing a laser capture microdissection approach, go in and also identify which are those gene signatures, which can again really also be used and integrate them together with that imaging mass cytometry data to again really understand how you can use these technologies to go in and predict new prognostic biomarkers in a tumor section like this one over here. So with that being said,
sorry, Roberto.
Yeah.
Sorry to interrupt. Can I please ask you to to wrap up the presentation?
Yeah, this is the last slide. No, absolutely. I was I took a bit more. So this is fundamentally the last slide. And in case you have any questions, you can feel free to reach out to me or to the local Oxford sales reps from Fluidigm, which of course are Tushar Shah for everything genomics related and Jonathan for everything Cytoph related.